seen what happens with the antenna complex. So now we're just going to put it all together. In this video, we'll talk about what a photosystem is, right? And essentially, a photosystem is sort of a large protein complex. And in that protein complex, we have our chlorophyll molecules embedded within it. And of course, these are what the light is hitting. And there's the chlorophyll B's, there's the chlorophyll A, and then there's the electron acceptor sort of embedded in there as well. And we know that what happens when light hits this complex, it causes electrons to become excited. And those electrons that are excited, if it was just chlorophyll in a test tube by itself, they would jump up a level, jump back down, release some heat, and maybe glow in the dark. But here, in this complex, the electron energy can bounce around and be passed eventually to the chlorophyll A, which then can give its electrons to the electron acceptor. And that electron acceptor is eventually going to give those electrons to something else. Those electrons are going to be given away to another molecule, which means that this molecule or this complex now is short a few electrons, right? It's missing some. And if you're missing electrons, that means you have a very strong desire to gain electrons. So once these electrons are given away, this becomes a very, very highly electronegative complex. And if anybody comes along, it's going to try and steal electrons. In fact, this complex is... all right, so if a molecule comes along, this powerful oxidizer, or electronegative complex, will steal its electrons. And as we're going to see, it's powerful enough to steal them from water, which is pretty tough, because now you're fighting with oxygen, right? Oxygen is trying to hold on to the electrons in water. This complex is actually going to steal from oxygen. That's how electronegative is. It's the strongest, most powerful complex, electronegative complex that we know of. The strongest oxidizer in biology, that is, in biological systems. Okay, so we call this the complex. And these complexes are proteins that are embedded inside the thylakoid membrane in the chloroplast. So now what we're going to do is we're going to draw a picture of the thylakoid membrane inside the chloroplast, and we're going to draw in these photosystems and see how they work together to produce a cascading flow of electrons handing off one to the other that we can then use to capture energy once again and do some work. So let's draw. Extend. All right. I think everything here is good. Okay, so let's draw ourselves a membrane again. Let's draw a little phospholipid bilayer. And then we have to put in some molecules. So I'll use purple. I don't know why that keeps popping up. I'm hoping it's not going to interfere with everything that's happening here, but yeah. Anyway, we'll just continue. We'll put this first one in here. This is called Photosystem 2. You'd be like, isn't it called Photosystem 1? Nope, it's actually called Photosystem 2. When we discovered the photosystems, we discovered this one second, but it turns out it's the first one in the process. So that's why it's called Photosystem 2, because it was discovered second. But it is the first one. And then we have to put more membrane, but before we do that, I'll take another color, maybe blue, and I'll put a little molecule right here. And inside that molecule, I'm going to write PQ. PQ, which stands for, we'll write the words down later, stands for Plastoquinone, which is a transporter. It grabs. It's the molecule that those electrons from the electron acceptor are going to be handed off to. Right? It's going to steal those electrons, those high energy electrons. Okay, so let's just sort of fill in around it a little bit more membrane, like this. Then we're going to draw another photosystem. This one is called Photosystem One. So we'll put a big 1 in it. And that's not working today, but okay. And then in between here, we're going to put another little complex. I'm going to put it in blue, right sort of here. And this is a cytochrome complex. It's a cytochrome enzyme that's going to be sort of grabbing some electrons. So let's just call that cytochrome. I'm going to call it cyt. And we'll put the other things in later. Then, over here in blue, I'm going to have another molecule. This molecule will put an F inside. He's called ferredoxin. We'll get to him in a minute. And then one more little blobby molecule sort of over here. And whoops, smart board. And this is Nadp Reductase. Nadp plus, actually. I'm going to have to get a smaller plus. And then reductase. Reductase. And then we'll fill in our membrane sort of all around. La 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 la. Like this. Put a few tails in there. For all the carboxylic acid tails. Okay, so this is our membrane. And then we can put some more membrane on this side just to kind of close it all in. All right. Oh, we need one more molecule, which I'll draw in a different color because he's sort of in a different, he plays a slightly different role. I'm going to draw him down here, right here, and I'm just going to put a P on him. His name is plastocyanin. We'll get to him in a minute. Okay. So now here's the process. We're just going to go through the story. So light hits the chlorophyll. Zam. Light hits the chlorophyll. I'll just draw one. But we know that there's an antenna complex. Well, in fact, I guess we can draw it in. Okay? And there's the dark one for the chlorophyll A, and here's the little proton, or sorry, electron acceptor right there. Okay? So the electrons, now we have to show the flow of the electrons. So the color I'm going to use, which I've used all my colors now. Let's pick a nice color that will stand out here. Perhaps this orange will work. Okay? So what happens is the electrons get excited by the light, and they bounce, and they bounce, and they bounce, and they bounce until they hit here, and they bounce to here. Then they're handed off to plastoquinone. And that means now that this complex, this photosystem 2, is devoid of an electron. And it wants something, it wants this electron back. But it can't get it because it's not able to steal it back from plastoquinone because of the electronegativity differences here. So it has to get it from somewhere else. And so where does it get it? Well, along comes a water molecule. Water that happens to be in the thylakoid comes along, and electrons are transferred from the water. Actually, two of them, if you want to, 2e, are transferred from the water, and that produces 2 H pluses and a half O2. Now, of course, that's just a chemical description at the lowest level. What's a half O2? Well, you can't really have a half O2. Basically, what would happen is this process would happen twice, right? And you'd end up with 4 electrons that were transferred in, and the remaining oxygens would then form a full O2 molecule. So it's actually 2 waters that would give away 4 electrons. That would make 4 hydrogen pluses and the full O2 molecule. But I like to show it this way because it's in the simplest form. And now we can see how photosynthesis produces oxygen. Question. All right, so now we have this electron that is passed to plastoquinone. Plastoquinone is a molecule just like ubiquinone, which can move through the membrane because it's non-polar. And so it can slide over to the cytochrome complex, back to my orange color, and deliver those electrons. So the molecule actually moves. It's not the electrons that go, it's the actual molecule, but for the sake of this picture, I'm just drawing it as the electron path. They go to the cytochrome complex. Okay? In the process of doing this, as plastoquinone moves, something cool happens. In order for plastoquinone to do this, because it picked up 2 electrons, remember? It picked up the electrons. So guess what it can do with them? Just like those electrons can be stolen from water and break a bond, now that plastoquinone has them, it can use them to make a bond. And so what it'll do is it will grab an H plus that happens to be here, and it'll grab that H plus, and as it's going through and hands off to the cytochrome, it will then move the H plus through the membrane to the other side. So in the process of handing that electron off to cytochrome, first it picks up the H plus because it's got the electrons, but then when it gives them away, it has to break that bond again to give them away, and the H plus is now moved. Does that make sense? Very straightforward, but very critical. Okay, then the electrons are handed to plastocyanin from the cytochrome complex. And then the plastocyanin transfers the electrons to Photosystem One. Photosystem One is almost like Photosystem Two, except the chlorophyll that are in it are sensitive to a slightly different wavelength of light. Photosystem 2 is sometimes called Photosystem is it 680 and 700? I think it's 680, yeah. And then this Photosystem 1 is called 700 because the wavelengths are slightly different. That's not really critical for us right now. It's the path. The orange is the path of the electrons. Okay, it's where the electrons are being handed. And then we have this photosystem again. This is almost the same thing, except it's sensitive, as I said, to a slightly different. So you see, those electrons are kind of spent, right? They've been handed off to plastoquinone, and then they've been used to make a bond. Then that bond has been broken as it hands off to the cytochrome complex, and then it's been handed down to the plastocyanin. So by the time it gets there, they've lost a lot of their energy. And now they're really not much good to anybody. So they're given to Photosystem 1. Guess what Photosystem 1 can do? Well, light hits it. Light hits Photosystem 1. Those electrons then are now given a second boost of energy, a second time. And they bounce till they get to here. They go to the acceptor, and now they're handed off, instead of to plastoquinone, they're handed off to this ferredoxin molecule. So let's put back to our orange. They're now handed off to ferredoxin. But now they have another boost of energy because of the extra sunlight. So it's like the same process that happened before, happening again, a double whammy, right? Yeah. Oh, you're right. This should be orange. It's the path of electrons being excited and bouncing around. So you're right. That should be an orange line if we're tracing the movement of our electrons with orange. Okay. Now you got ferredoxin, which is a molecule that can move through the membrane. And so ferredoxin then transfers the electrons to the Nadp reductase. So the electrons are handed over here, right? The Nadp reductase, you can kind of guess what it's an enzyme. It ends with A's. You can guess what it's going to do. It's going to take a molecule called Nadp, and a plus. Let me get a smaller line here. Nadp plus. This molecule is floating around. We haven't been introduced to him yet. He's very much like NAD, but he's got a little P on the end. He's got some phosphate attached, right? So he's going to slide in, and he's going to grab those electrons. He's the last one who's going to take those final electrons, which I'll now go back to orange. So the electrons are going in here. And of course, that means he can make a bond. He can use those electrons to pick up an H+, which happens to be floating around. There's H+ everywhere, right? And so that means he can become NAD. Let me write it in thinner. NADPH. Sorry about the crazy smartboard, but that says NADPH. I'll try it again. I don't know what's going on today with technology. Maybe if I don't press so hard and just go easy. Treat it with really tender, loving care. Just like people, when people get stressed out, you have to treat them a little more gently. There, that's a little better. NADPH. Okay? So what we should also do maybe is put NADPH and an H+. Put that going in there as well. Or you could write NADP plus H+, right? Whatever you want to do. And now we have this molecule, NADPH. That molecule is going to be useful later on. We've constructed it. We have used the light energy from the incoming light to excite electrons and cause them to follow this chain of passing on from one oxidizer to the next, until finally their energy is handed to this NADP molecule, and now it's holding that energy in the bond with that H. And that molecule is going to go off to the next part of the photosynthetic process, which we call the dark reactions. Ooh, because they're dark. They happen without light. They happen all the time, but they happen without light. And so there we have it. But there's one more piece to this puzzle. Okay, so we want to just summarize some of the key things that are resulting from this process. You'll notice that key thing number one is the production of NADPH. Okay, so the first thing we want to sort of summarize or look at here is NADPH being made at the top of the right of the picture in green by this complex. In fact, it's the final resting place of those electrons. Those electrons end up stuck in that hydrogen bond, or that bond of hydrogen to this NADPH. They're right in there. That's where those electrons went. So water's 2 electrons eventually end up there because of all this excitation. The other thing that's happening that you should notice is, in order to make this, we have to use up H+. So that means we're using up H+ in the top part of this. We're also pumping H+ right here across. So that means we're creating a concentration gradient. If we're pumping it here and using it up here, we are getting a very low H+ concentration on this side of the membrane. And over here, because it's being pumped, we're getting a higher H+ concentration. So we are also generating at the same time an H+ gradient across the membrane. Yep. Okay, so look what's happening on the top. On the top, H+ is being used up here. So it's gone, it's being taken, right? Hi. Okay, so this means that H+ is being reduced in the solution. It's lowering, because it keeps getting attached to the NADPH, or the NADP. Also, at the same time, see the H+ here? It's being carried through by Plastoquinone. So that means there are 2 drains. There are 2 mechanisms that are draining H+ from the top, right? But where is it going? It's going down to the bottom. So we're filling up the bottom with H+, making the concentration higher on the bottom and lower on the top. Sorry? Yeah, it's being carried or pumped to the bottom by the plastoquinone, right. Now, you'll remember that if we have a hydrogen ion concentration that's different on one side of a membrane or another, we can power an ATP synthase complex. And so just like in the electron transport of respiration, there's another piece to this puzzle, which we're going to put in. I'm going to do it in a separate video, but there's another protein complex here called the ATP synthase, and his job is going to be to utilize this H plus gradient, and guess what he's going to make? ATPs. Okay, so let's draw, we'll do that in the next video. We'll talk about how the ATP synthase works. Actually, yeah, we'll do another video just for kicks. That's enough for this. So there's the process. You have to be able to identify the players. You have to be able to understand what these photosystems are made of, these antenna complexes of chlorophyll A and B and electron acceptors. You have to be able to sort of understand the path of how the electrons are transferred from one to the other. You have to understand plastoquinone pumping H+, and you have to understand the NADPH being made and how those two things affect the concentration of hydrogen ions and create the gradient. All right? We'll call that a day.